Hello class. In the last lesson we talked about the four different kinds of macromolecules. We talked about proteins, carbohydrates, lipids, and nucleic acids. We talked about their structure and their function. In this lecture we're going to focus on one of those four. That is to say, we're going to look at proteins today. And in particular, we're focusing on a particular subclass of proteins known as enzymes. Okay. Many of us should know what enzymes are from our sophomore year. But we're going to be talking about enzymes because in the next couple classes we'll be involved with a couple laboratory investigations about enzymes. We will be simulating the function of enzymes next class, and on Friday we'll be actually going into lab and working with a particular enzyme known as catalase. Okay, so it's important for us to know what enzymes are, how they work, in order for us to be able to investigate in an educated fashion. All right, enzymes are protein catalysts, and we're going to talk about all of that significance in the upcoming slides. However, before we get into a discussion about the function of enzymes, we first have to know that enzymes are proteins. All right, and I'm going to underline that one more time. They are proteins, all right, and they operate, or they are structured just like any protein. All right, if we take a look at the enzyme, or rather the protein that is pictured below on this slide, we'll see something significant. We see this protein is a long-stranded molecule, but it's a long-stranded molecule that has a very characteristic folding pattern. You'll notice that there are actually two strands of peptides. One strand is red, right, over here, and the other strand is green. And you'll notice that both the red and the green strand, they look like they're crumpled, or they are folded. And some of those folds are in spiral fashions. Some of them you might carefully look at are in sort of zigzag fashions. But the point is that enzymes are proteins because proteins are always folded molecules. They are characteristically folded molecules. And sometimes one folded strand can bond and interact with another folded strand. We call these two strands different subunits. All right, let me write that. We've got subunits here. So you'll see that there's a red subunit and a green subunit. We'll be talking about subunits in a bit. Again, let me reiterate that because enzymes are proteins, enzymes, like all proteins, are folded. But let's ask ourselves the question, why do proteins fold? Well, we should know by now that proteins are long strands of amino acids connected together. When I say long strand, I'm really saying that a protein is a polymer. Right? It is a long chain-like molecule. All right, and it's a polymer of different monomers. Those monomers are amino acids. All right, so we see a bunch of amino acids bonded to one another. Okay? We call this the primary structure. Oops, I just crossed it out. We call it the primary structure of proteins. The primary structure is the sequence of amino acids. But we know that amino acids can bond with one another. Some of those bonds take place at a relatively short distance. We call those short-range interactions between amino acids. The short-range interactions are the secondary structure of protein folding or protein structure. The short-range interactions can take the form of beta, that's a Greek letter beta, a beta-pleated sheet, or it can take the form of an alpha, another Greek letter, an alpha helix. Okay, an alpha helix looks like a spiral, and a beta-pleated sheet looks like a folded piece of paper, for example. And those short-range interactions, or those beta-pleated sheets and alpha helices, also can interact longer distances away from those short-range amino acid interactions. That tertiary structure, the third level structure, is the long-range interactions between amino acids. Right? An amino acid at this part of the strand might bond with an amino acid way further down the strand and make that protein strand fold in characteristic globular fashion. So we'll say when the protein is crumpled up looking like something like this, we call this the globular form. okay, the globular form of the protein structure. And so we see the tertiary structure is the globular structure because there are long-range amino acid interactions. And then the final level of protein structure is known as the quaternary structure. The quaternary structure occurs when one globular strand of amino acids interacts with a second or third, or in this case, four different other globular structures, or four different other tertiary structures of amino acids. So we have four levels of protein structure. The primary structure is the sequence of amino acids. The secondary structure is the short-range interactions between amino acids, short-range bonding. The tertiary structure is long-range bonding that creates the globular structure of proteins. And the quaternary structure is when we take that globular structure and interact it with other globular structures or other strands of folded proteins. So we've said that proteins are folded molecules, and we say that the folding results when amino acids are bonding with other amino acids. And usually they're bonding via hydrogen bonds. Sometimes they're bonding through what we call hydrophobic interactions. Right? Hydrophobic interactions. That's when a hydrophobic molecule will interact with another hydrophobic molecule. They can even bond through ionic interactions or ionic bonds. Okay? The question is why? Why do amino acids want to bond with one another? Well, we have to look at amino acids again. So let's take a look at those 20 amino acids that exist. The key to understanding anything that deals with protein structure is to understand amino acids, and specifically to understand the R group. All right, remember the R group, because we have to remember that the R group is the group on an amino acid, or it's the side group of an amino acid that can change from one amino acid to the next. And we have to know that some of those side chains or some of those R groups are nonpolar R groups. Okay, we take a look at some of the functional groups attached. All right, we see some nonpolar or hydrophobic R groups are simply a hydrogen or a methyl group or a number of methyl groups or even more methyl groups or more methyl groups. Those methyl groups are nonpolar, and because they're nonpolar, they will be hydrophobic. Okay? Again, we have to remember functional groups to make any sense of this. Some of those R groups, instead of being nonpolar, are in fact polar. We see that any amino acid whose R group contains a hydroxyl group, and any hydroxyl group will make the amino acid polar. We see a sulfhydryl group in cysteine. We see, for example, a ketone in asparagine as well as glutamine. Right? We see another ketone. Okay, so we see polar side chains, and with two polar side chains, we can generate some hydrogen bonding. Okay, if I can spell properly. And of course, we have at least 5 amino acids whose R groups are not nonpolar, nor are they polar, but instead they are electrically charged. That is to say, we have a carboxylic acid and another carboxylic acid to generate negatively charged R groups. Or we have an amine group here and another amine group, or some other kinds of amine type groups to generate some kinds of positively charged side groups. And when we have a positively charged side group coming into contact with a negatively charged side group or R group, we're going to have some kind of ionic interaction, because two ions of opposite charges will attract one another. So we can now understand why amino acids would ever want to bond with one another, and as a result we can understand then why proteins might fold because of the interactions between and among the amino acids. So take a look. And in this instance over here, we have a hydroxyl group and a ketone, and we're going to see some hydrogen bonding between those two polar side groups, or those two polar R groups. We see some a nonpolar R group with another nonpolar R group folding in some kind of hydrophobic interaction. anytime we have cysteine and we have two sulfhydryl groups, those sulfhydryl groups will form bridges. We call those disulfide bridges. So we see a bridge, right? And those bridges can form bonds between two cysteine molecules or two cysteine amino acids. And finally, we see an amine group and a carboxylic acid coming into ionic bond with one another. All right, again, so we can now understand why proteins and amino acids would fold given their primary structure. And we can then infer that if we change an amino acid, for example, if we change our polar amino acid into a hydrophobic or nonpolar amino acid, that will change the bonding structure. So in essence, if we change the primary structure, that is to say, if we change an amino acid, we will change the secondary structure. That is to say, we will change the short range or the short range bonding, like beta pleated sheets or alpha helices. And if we change the secondary structure, we're going to change or alter the tertiary structure, and hopefully or eventually, potentially even the quaternary structure. So changing primary structure has far-ranging impacts or far-ranging implications towards the entire structure of and folding pattern of any kind of protein. Having an understanding of protein structure will help us have a deeper appreciation for enzyme function. But first we want to talk about what enzymes actually do. So let's take a look. The question, of course, is what do enzymes actually do? Ultimately, the answer is our enzymes act as catalysts or biological catalysts. Now, a biological catalyst, or any catalyst, is something that speeds up a chemical reaction, for example. So it speeds up a chemical reaction. In order to understand better how it speeds up chemical reactions, let's even step back further and talk about chemical reactions. At its most fundamental, when we say something is reacting chemically, all we're saying is that we take, for example, substance A and substance B, and we smash them together and they form some kind of chemical bond. Okay, we've made a bond. Okay? Or we take chemical AB and we heat it up or give it some energy and we break that bond so that we have chemical A and chemical B. All right? So what we've seen here is a chemical reaction is anything where we make a bond, anything where we break a bond, or we might have chemical AB plus chemical C, and we would break this bond between A and B and form a bond between A and C, and we have a third independent or isolated chemical B. Or we see chemical AB and chemical CD, and we break bonds between A and B and between C and D, and we rearrange and make new bonds between A and C and B and D. To put it simply, chemical reactions are anything where we break bonds, where we make bonds, or where we rearrange bonds. And rearranging bonds involves breaking one bond and making another bond. Ultimately, chemical reactions are bond breaking, bond making, and bond rearranging. However, chemicals or molecules don't just react chemically out of the blue. It doesn't happen spontaneously. All the time, any kind of chemical reaction requires some kind of energy in order for that chemical reaction to take place. In order for that chemical reaction to activate, we need to input some energy. Activation energy is energy that is needed to begin a chemical reaction. That is to say, any energy that is required to make and break chemical bonds. Again, like I said previously, chemicals or molecules are just bouncing around, and they will make or break chemical bonds if they're bouncing around with enough energy and they're moving quickly enough so that they can bounce into one another and make a bond or break a bond. So we need some activation energy in order to start any chemical reaction. Which brings us back to enzymes. Enzymes are catalysts. They're catalysts because they speed up chemical reactions. And they speed up chemical reactions because they take the activation energy that's normally required to start that reaction, and they lower that activation energy. All right, notice that we see this amount of energy is the amount of energy in an uncatalyzed reaction. That is to say, in a reaction that would normally take place spontaneously, it requires that amount of energy. All right, that change in Gibbs free energy, change in G. However, with the catalyst helps facilitate, helps speed up that reaction because it only requires a lesser amount of energy in order to start that chemical reaction. And if it requires less energy to start the chemical reaction, well, that means that it can start the reaction quicker and finish the reaction faster. Thus, the reaction will take place more quickly and it will have sped up. Now, it's great to say that enzymes speed up chemical reactions by lowering the chemical reaction's activation energy. But that doesn't mean much unless we understand how the enzyme's structure plays an important role in the enzyme's function. So we have to revisit amino acids and protein folding. We already know that a protein's primary structure, or its sequence of amino acids, predetermines the folding pattern of that protein. In enzymes, the folding pattern occurs in such a way that a pocket is formed in the globular form of that enzyme. The pocket in an enzyme is called its active site. And an active site is the place where a substrate binds. What you can see here in this diagram here is that this enzyme's active site has a very characteristic shape. In fact, it's a very specific shape. It's a specific shape because it only allows the binding of a single or particular substrate. As a result, we say that enzymes' active sites are very specific. There is a great specificity in an enzyme's ability to bind with a substrate. There usually happens that only one substrate binds to one particular enzyme. As a metaphor, we can understand an enzyme's function is to be something like a matchmaker. Its job, in many ways, is to take a particular molecule or a couple of molecules that we'll call substrates. It'll take those substrates and put them in the same place together, put them in the same room together, because those substrates fit very well in that enzyme's active site. Once the substrates are in the enzyme's active site, the substrates are held into that site with some weak interactions. And because the enzyme configures those substrate molecules in a very specific fashion, it can lower the activation energy and facilitate bonds being made, or bonds being broken, or bonds being rearranged. In this case, we see two substrates of particular shapes and particular colors, for example. We see them fitting very nice and tightly or very well into our enzyme. And in this stage, we call it our enzyme substrate complex. The activation energy is lowered, bonds are made, bonds are broken, bonds are rearranged, and the substrates, those reactants, are then converted into products. Notice they still have the same shape, but certainly we can understand bonds to have been rearranged here. And once those bonds are rearranged, the products, what used to be the substrate, are then released back out into the outside of the enzyme. And what we'll see then is that enzyme will go back to its original shape, back to its original conformation. It's ready then to use some new substrate. Notice that the enzyme itself is not used up. It is not used in the chemical reaction. It just facilitates that chemical reaction. So we can use the same enzyme over and over and over again, and that enzyme will continue to act like our matchmaker, facilitating the putting in the same place two substrates that maybe or probably wouldn't find themselves together. If we can understand that some enzymes act as matchmakers, Sometimes all other enzymes decide that instead of making the match, they will instead speed up the breakup. For example, if a disaccharide like sucrose is the substrate for a particular enzyme, such as enzyme known as sucrase, that substrate will fit very well into the enzyme's active site, and that enzyme will facilitate the bond breaking or the breaking up of these two monosaccharides. All right, and so we see some bond breaking, we see conversion into products, and we see those products released back out into solution. Again, that enzyme is not used up in the process. All right, we get to reuse that enzyme as often as we'd like. Again, the bottom line is this. An enzyme is a protein, and like any protein, an enzyme is folded. It's folded in such a way that it forms a pocket or an active site in the globular structure or the globular form of that protein. That active site will bind with great specificity any substrate that can fit well, and the enzyme will speed up or facilitate bond breaking or bond making. We know that this shape is determined by the folding pattern, or the function is determined by the folding pattern. The question is, what if we unfold an enzyme? What happens to that enzyme? Let's take a look. If an enzyme's function is predetermined or determined by its folding pattern, or in other words, if a substrate can only bind to an enzyme if the enzyme's active site is in a particular shape, if we change the shape of that active site, or if we unfold the protein or enzyme, we will take away that active site. All right, as I move around our page, we will remove that active site. Will be destroyed if we start to unfold that protein. And if we destroy the active site, that substrate will not be able to bind to that enzyme, and that enzyme will not be able to speed up a chemical reaction. All right, we destroy the enzyme's function if we destroy the active site, and we can destroy the active site if we unfold the protein. There are many ways of unfolding the protein, and all of those ways we call denaturation. Okay? We denature enzymes if we unfold those enzymes. All right? And so we lose biological activity. We can do so by changing the pH, because most proteins operate under ideal pH conditions. Most enzymes don't want low pH or high pH. They want some kind of neutral or buffered pH. If we change the temperature, with an enzyme, we might add too much heat so that those amino acid interactions, the polar interactions, or the nonpolar interactions, or the ionic interactions, they may be exposed to too much energy and stop bonding with one another, thus unfolding the protein. If the ionic strength is too much, for example, if we add salt to a solution full of enzyme, the ions from the salt can interact with the amino acid side groups and change the folding pattern and destroy the active site. And those are all sorts of things that can denature a protein. But if we remove these things, for example, if we renature, if we bring the pH back to a normal level, or the temperature or the ionic strength back to a normal level, we can renature an enzyme and refold it so that its function is recovered. All right, so this lecture is all about enzymes. It's all about the structure of enzymes and how that structure facilitates its function. That is to say, how the primary structure and tertiary structure of an enzyme helps it speed up chemical reactions by lowering activation energy. All right, if you have any questions, send me an email and we can get to it.